to the Weekly Bioanalysis, a KCAS podcast.
Hello, and welcome to the 89th episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO servicing the pharmaceutical and biopharmaceutical industry for over 40 years. My name is Dominic Warrino, and I've been in the bioanalytical drug development space for a little over 30 years with a focus on biologics. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LCMS space for around 30 years.
John and I are members of the growing scientific advisory team here at KCAS Bio. Either or both of us are available to answer any of your questions you may have regarding this podcast or any of KCAS Bio services. Today we have a special guest, Jeff Goddard, our Senior Vice President of Corporate Development, will be joining us for an action-packed main topic discussion. We are thrilled to have you listening to the 89th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here, and we are looking forward to a fun episode. To kick us off for podcast 89, John is going to go over today's podcast topic a bit before we jump into the news and resource section. John.
Hello, and yes, this should be a fun and different discussion. We wanted to invite Jeff on to talk about the cake. We've been with KCAS for a while, and the company has changed a lot in the time that we've been here. So we want to talk about the KCAS strategy for balancing growth with a good employee experience, and this will be the first part of a two-part approach with the other guests joining us next time. But we want to just talk through how KCAS has grown as a company.
Okay, great, John. Let's kick us off with our first news item.
Let's start with antiviral chewing gum might revolutionize how we tackle infectious diseases. This is an interesting strategy to possibly tackle common viral infections, such as flu, that cost the US an estimated $11.2 billion each year in lost productivity, which burdens both our health system and the economy. Also, another virus that transmits through oral contact is herpes simplex virus, HSV1, which infects over 2/3 of the global population and can, in rare cases, lead to encephalitis or infectious blindness. Now, for all these things, we usually think of vaccines as the first defense, but as you know, if you read anything in the media at the moment, there's a lot of loud anti-vaccination voices. So, alternative strategies to tackle these kind of viruses is a good idea. So researchers from University of Pennsylvania School of Dental Medicine, in collaboration with a Finnish team, are looking at using chewing gum infused with antiviral compounds. They published a study in Molecular Therapy, and it showed that the gum can actually significantly reduce the viral load in the mouth using compounds naturally found in beans. A couple of the candidates they used, the CTB ACE2 to gum, which neutralized over 95% of SARS-CoV-2 particles in the saliva of COVID-19 patients, including major variants like Beta, Delta, and Omicron. And so they're actually looking at moving forward into clinical trials with this. And then another, Henry Daniel, a professor at Penn School of Dental Medicine, also used FLT3 receptor interacting leptin, which came from lab beans into gum and looked at treating both influenza and HSV. I'll pass it to you, Dominic, and I'll talk about some of the mechanics of this, because it's sort of a fascinating.
It really is.
And also how they did it, and it's like, yeah, and where they want to go is also, oh wow, these people are really on fire.
Yeah, no, John, I studied vaccines, right? Part of that studying back in like 2000, people were trying to add, certain elements to potatoes to help with creating a vaccine for HPV. So I studied HPV. And so the concept of eating or feeding a vaccine isn't new. It's just it wasn't that effective back then. There was a variety of reasons, and the biggest reason was the mucosal immune system, right? From your
Hmm.
mouth to your anus is your mucosal immune system. It functions differently than like your serum plasma, circulation does, although they do coexist, but it has different cell types. You can imagine it's got a very different environment almost. And gut immune's all the rage, right? We've heard about all that. But the idea, this working is it's just fascinating. And the other part is everything to cure us is out there naturally. That's what this
Hmm.
tells
Yeah,
us.
yeah.
It's just hidden somewhere. We just got to figure it out. But I still can't. And they touch on it. I'm going to pass it back to you in a second. They touch on how part of the viral entryway is the mouth, right, and the nose. So it makes sense if you could treat it there. I'm still this reading that we got here doesn't talk about how they overcome some of the gut immune stuff, but maybe it's just because of this. What is it? FLT3 lectin-like protein is absorbed differently. It says lectin right in it, which could be because those are absorbed differently. But because most viruses, when you think of, excuse me, most vaccines, right? There's some sort of like we have virus-like particles, or it's a protein. All that stuff just gets immediately kind of degraded as it gets into your gut and things like that. Where a lectin probably has a better, not probably, obviously it is effective in some capacity.
Hmm.
John, this is revolutionary,
Yeah,
right?
I,
Like,
I think I mean
give me
it
a
does.
little tablet. It's like quitting cigarette smoking.
Yeah,
Pass it back to you. It's
I
fascinating.
think as it points out, I mean, sorry, I'm yeah, I misread something. So if you want to keep going, I'll I think this is like I say, this is looking at. It's not curing it, but it's really cutting the load. So
Mhm.
it basically tamps down on transmission. I think is the key here. I like I mean, they looked at you know using a mastication simulator, mechanical tune device. They showed that over half of the active compound was released in 15 minutes from the gum, and just 40 milligrams of the FLT3 receptor interacting lectin was enough to reduce more than 95% of the viral load present. I love the fact that they're looking also at testing against bird flu. So the obviously the chewing gum isn't going to work for birds. That would be quite amusing to watch if it was. But then it's looking at this powder, the lab bean powder, to see if they can add that in bird feed and inhibit it through that approach. So I just thought like I say, it's a very interesting, elegant. Complement to vaccines, and it just another possible way of targeting some of these viruses and controlling them within the population.
Yeah, it would be funny to see the bird chewing, but it's actually really when you think about cost savings and some of the
Hmm.
issues we've had with bird flu viruses and things like that. But putting vaccines into feed isn't new. There's a whole company
Sure, yeah, yeah,
that does that.
yeah.
I think Siva does it with chicken. It's the Newcastle vaccine, I think, is in their feed, and there's a variety of other feeds they give animals. But John, it shows that basic research, right? I'm kind of putting it in that bucket, which we don't. We're on the drug development side, right? Pharma companies and biotech companies aren't really in what I would think of as more this basic. But this is, it's near and dear because basic research is where we get all of our, kind of um, solutions or future
Hmm.
drugs from.
Yeah, yeah,
You can move on, but this
yeah.
is this
You're
is
right.
really, really cool.
I
One thing,
think,
one interesting thing though, John, is think about this. Do you have to chew the gum all the time? So it makes sense. Look, the
yeah,
entryway to the mucosal immune system. So this like lab bean powder extract, whatever it is. So you're chewing it, it gets released. That's where like, what's the kinetic of that? Does it get into the like? They don't really go into details
yeah,
there. It
exactly.
is interesting. Do I have to chew a piece of gum monthly or whatever? But still, I think it's just first step. I mean,
Yeah.
I think it's great, and it kind of perked me up a little bit.
Yeah,
This is
you're
really
and
cool.
you're right. To go back to the basic research thing, I mean, we work with pharma companies, but that's not where a lot of the basic ideas originate. They have the ability to apply, very directed, building on the ideas
Yeah.
that comes out of universities and other research groups, and that's the research engine that drives the pharma industry. It's and this kind of thing. You got universities saying, here's the basic idea, and we always see that mechanism where a professor has an idea, might do a startup company, but at some point they need investment from outside, and a lot of them get so far and then they're bought out by large pharma, and that's when things make it to market. So yeah, I mean, there's a definite process here, and the basic need for basic research is huge to feed the entire drug industry.
I'm sure
Anyway,
the
moving.
the
Sorry, go on.
basic research is being done at their incubators and things like that. But it's a fascinating concept. But it looks like Jeff might have something to add. Jeff, what do you got for us? Go ahead, Jeff.
Well, thanks. I was just, if this works, it's just really, I just picture we all were, masking up like crazy when COVID came around, and all these sorts of debates on the science behind it and working or not working. But I get on the planes a lot, and instead of masks, if they handed out gum like this while you're stuck in a tube everywhere, and it just helped prevent transmissions, it'd be amazing.
That could be it, Jeff. You're absolutely right.
Based, like,
yeah, sorry, Jeff, go ahead.
Yeah, no, just as a population based sort of like flu season, daycare centers where, little cesspools of infectious disease. It'd be great if gum could sort of help stave off some of these localized outbreaks a little bit.
Yes, certainly as a intermediate solution, or short to intermediate solution, until the longer term solution actually is made available, because we had a long period obviously where we had no vaccine at all for COVID, and it was had a devastating effect on the population.
Yeah, no, I think you both bring up great points where it's not a cure. It's not like something that can absolutely prevent it like 3 jabs in your shoulder. I think you're right. It's to prevent the transmission via orally. And what a fascinating concept to like you said, daycares or planes or whatever it is. And even John, again, I just want to rehash what you said. You're right. If maybe there's some immediate relief, right? Even if you could cut down the next pandemic by providing, chewable bubble gum for everybody. But
my parents would be horrified if they saw me chewing gum on a daily basis to prevent an illness. It's like the years that they tried to drum into us. You shouldn't be chewing gum. It's a terrible habit.
Yeah, there'll be gum under every elementary school desk again. But let's move on, right? Like,
yep. Next, this is something I think is really. It's one of those like throwaway articles, but actually is quite fascinating the more you think about it. J&J finds pace of oncology innovation is overwhelming physicians. And so this is basically Johnson & Johnson has generated evidence that pace of oncology innovation is overwhelming physicians. In a recent survey that the company commissioned, oncologists are struggling to keep up with new treatments and guidelines, pointing to a need for additional support to ensure patients get the most appropriate therapy. And I was thinking about that. So from a bioanalytical point of view, we see all these different kind of treatments, and it's fascinating. We get ADCs come in, we work on those. We have small molecule, anti-cancer compounds. We have like large molecule, all these different approaches to try and attack different cancers, in terms of blood, solid tumors, etc., etc. So I'm not surprised that someone downstream is feeling a little bit overwhelmed as these things come through. And so, that was what struck me about this. It's like, we sort of enjoy all this variety of what's thrown at us in terms of different things to work on. But for the end user, I mean, all these different, different entities with the different approaches and different, side effects, etc., etc. How do you get all the information you need to ensure that you're prescribing appropriately when it comes to these different agents?
Yeah, it doesn't surprise me at all. It's a little bit disappointing, but if you've, having spoke to the pediatrician and even my own physician, they're kind of in their own little hemisphere, so to
Sure.
speak. They're not really thinking about innovative medicines, and even just friends sometimes will ask me questions if they have an individual in their family that's by getting therapies and stuff, and I'll throw things out, and they're like, "Oh, my physician had never heard of this," and it's just with all the AI we've got, with all the technology we've got, how come our physicians are so far behind? But maybe we'll move on. That's what it feels like to me. How do they not have the fingertips to do it? But I'm not a physician. I've
And,
spent
and,
rotation
and
in the ER, and I didn't like it at all. I don't like hospitals. I don't like the smell of them. God bless our physicians and nurses and all
I
that.
know, but I wonder if it's the mechanism of how are they educated with these new things coming out? Because when we see, I know um a new treatment is approved, the next thing you see is that drug company building up their sales team. So although you got that new product coming on market, the physicians are having to deal with people selling them a drug.
Yeah.
So it's like that's not really education.
Uh-huh.
That's saying we have this product that's going to do this, but then another company says, but we have this product that can equally do that. And so how do you choose what's best for your customer? And I think that's possibly where the education piece is falling down. I mean, this is basically saying from this survey, it suggests that continuing medical education helps, with 92% of oncologists agreeing it is crucial for providing cutting edge treatments, but that more support is often needed. So extra support could help close the gap between the availability of new drugs and their successful implementation in clinical practice. I say, I just thought it was one of those off the cuff articles that I thought actually was quite interesting in terms of the underlying issues there.
Yeah, I'm not saying you should go out and listen to WebMD or whatever it is, but it's concerning when 70% of the oncologists admit they struggle to navigate it, but 92% say they want it, right? So that's
Yeah.
it's a big gap there in ability to get medicines to patients. But then you're right, it's like the squeaky wheel gets the oil, right? So if you have a big sales team, then that drug's going to sell, right? Shouldn't have to be that hard.
Mhm.
It's
Yeah,
an
it's a challenge for the industry as to how best we're innovating away. How best do we then educate the end users that as to what's the best option for their patients? Because ultimately, what we're doing, we're developing drugs to help people, have longer lives or cure whatever their condition is. And if we can't educate the doctors, then that can't happen. So we've hit a dead end. So the implications are big on something like this.
I have to admit, the clinicians, physicians, and things like that, they're resistant to some of this, at least.
Sure.
And I think that there's an aging physician population that probably is even more adverse to this. And we're one of the few countries that still start with surgeries and radiation, where a lot of countries don't do that. I know I took a left turn there, but think about it. Maybe part of the reason why we don't have as many, immuno-oncology, or however you want to think of it, therapeutics, is because they just radiation surgery. That's kind of the first line that we do here, where a lot of other nations, because they don't have the resources, they turn towards medicinal ways of
Hmm.
trying to
Yeah.
treat.
Yeah.
And so it's a fascinating. I've often felt like, if as an immunologist, if you do the radiation and chemo and it works, well, that's great. But if it doesn't, then that thing's really aggressive and it can be dangerous. And a lot of people say you actually can. Well, not a lot. I'm going to stop there. Those are my own opinions. I want to be clear about that. But it's a nice philosophical discussion. I would love to sit over a beer and share
Sure.
more of my thoughts about how we treat each other, and all that great stuff about health. But maybe we'll move on,
Let's
because
move on, and
that was a fascinating. That's eye popping to me.
we'll talk about someone else treating themselves.
There you go. See, that's more up
This,
my line, right? I'm going to be in my
yeah.
garage making dendritic cell therapeutics.
So I've revealed my obsession with snake venom in the past due to
Yes, you
some some really enjoyable work I did when I was at NIHS in North Carolina. Um, this is 18 years of self-inflicted snake bites lead to new antivenom. A guy called Tim Freed in Australia, 18 years ago, began willingly injecting himself with escalating doses of venom from 16 snake species. Now, antibodies in his blood have led to the development of a three-part antivenom cocktail capable of protecting mice from toxins of a variety of dangerous snakes. Now, these are all of the class Elapidae, which are cobras, taipans, and mambas. In this country, the coral snake is part of the same family. And so it's basically bigger picture is this. Even the proof of principle is that even a limited cocktail can actually have broad recognition of many types of related snake venoms. So in this case, the elapids. Where this doesn't address is the adder family, or the vipers, which is the other big pardon, the big snake family. So The antivenom cocktail, contains 2 antibodies isolated from Fried's blood and was tested against a panel of elapid venoms. Because Fried's body had been continually exposed to venoms over many years, his immune system has been able to fine-tune the antibodies it generates in response to snake toxins, a process akin to evolution by natural selection. I'm just changing the page here.
Yeah.
So the two antibodies. So in elapid venom, there's sort of short and long chain neurotoxins, and from a mass spec point of view, the long ones are around 6 to 8,000 molecular weight, and the longs were around 13,000 molecular weight. I tell you, I'm fat. I love this stuff.
You're just bragging a little bit there with those.
Yeah, I've got to call on my past. You do the same. You do the same when it comes to vaccines and a lot of other stuff. So anyway, that's the bulk of like cobra, mamba, etc. venom are these what we call small. Like polypeptide small proteins. And then the third part of the cocktail is actually varispladib, a compound already known to block the effects of another major venom toxin, phospholipase A2. And so I think I know we've touched on the past. With the most recent time we touched on this was a synthetic synthesis of
version of this, right?
Based from the Liverpool School of Medicine, and so that was one approach, because the point is a lot of what we have for antivenoms aren't very effective. So we're still trying to find ways of making this more effective, because they take out. We talk about the economic effect of flu and things. The effect of snake bites, although you don't know much about. It's huge, and I don't have the numbers. So it is really worth pursuing how better antivenom to hopefully be able to address all these accidental bites that happen to people just on their daily lives.
Yeah, it was a big number of people that die of venom.
It's here actually. Venomous snake bites kill more than 100,000 people every year. But it's not that it doesn't stop at killing. It's the disablement, because if you ever look at photos of bite sites and things like that, and necrosis of flesh and all sorts of stuff. I mean, snake bites are nasty things.
Yeah, especially if you're a little bit older.
Anything we can do to help it is a good approach.
Yeah, and there was a shortage of venom, and that's why they were making synthetic. And this individual who's doing this is just absolutely crazy, in my opinion.
Absolutely.
I don't recommend anybody self-dose themselves. Although, if you know the story of like H. pylori and how we kind of fixed acid reflux, that individual sat there and gave himself the H. pylori bacteria, and then took a Tums and cured himself of it. And that's why we got all omeprazole and Prilosec and all those things. So it's fascinating, but you got to be a little nuts, a little crazy. But it's just wigging me out. I don't like snakes. The whole thing is making me shiver a little bit. The thought of like, I don't know, but it is, it was surprising, 100,000, maybe that number is not as shocking, but you're right. There's a lot of people who get dismembered by this. And then like the lack of having it available, that was interesting to me, why there was like a supply chain issue with the snake venom. But hats off to this individual. Again, for those of you that are listening that might want to inject themselves with something to try and cure something, we highly suggest you don't do that.
Well, I think even Jacob Glanville, who's the CEO of Centivax and who is one of the people developing this treatment, basically said no one else should do what Tim did. We never gave him any guidance on what to immunize with, and he's done almost all of it before we met him. I think Tim did something remarkable, and there's a history of self-experimentation that's resulted in Nobel Prizes. But but on the other hand, I don't think this experiment ever needs to be repeated.
Funny. And it's like the flying cars are the wave of the future, but I don't want to be the one test driving those things, right? If flying suits and all this stuff, this falls right into that category.
Exactly.
There's a handful of people that might have won a Nobel Prize, but good Lord, the number of people that probably dropped dead from this is high. I bet. I wouldn't recommend it, but anyhow, it's funny stuff. And I don't know, John, you got one last comment because story is a great
I
one.
don't have any more comments. I just thought, like I say, it's fat. It's again, it's that sort of people. I'm always. It's always fascinating what people will do. I think, yeah, that Tim Fried, what he did in terms of treating himself, he must have had some goal in mind, thinking, I wonder if this will work. And I mean, God, what a high risk thing to do.
Well,
But it's
Lorenzo's oil comes to mind too.
yeah,
The guy trying to treat his son. So I mean, sometimes if you're in desperation mode, it makes sense. But I don't know. This is he's an Australian guy, right? He was in rural Australia. He probably had people in his life that had been affected by
I
life.
think
He thought, hey, I'm gonna maybe he knew he was naturally resistant. At some point, maybe I don't know. But it just goes to show you how powerful the immune system is, right? You can
yeah,
really
yeah, yeah.
augment it and prime it, right? All good stuff.
I think to me, I mean, I think and I think what sort of is interesting to me is that I wonder how this ties back into evolution. Because
Mhm.
obviously, if you've got commonality in venoms between the different snakes, it takes it back to the root of what was the
Yeah.
species they all evolved on. And I find that piece fascinating
That's
too.
deep, John. That's really deep, right?
Yeah. So I
From
mean,
the
I remember.
brilliant.
I'm going to touch on this because actually, with the work I did at NHS, because we were just playing with the LCM, capillary electrophoresis mass spec, but I mixed two venoms of two related species, and there was a lot of things were all very different. But even within that one, within those two overlapping species, there was one peptide that turned up in both of them, which
Mm,
was actually, and that was not a full identification. That was based on migration time and molecular weight. That was all the tools I had to hand. But it
mm.
said there was a possibility. I mean, that points to commonality, but you'd obviously have to do more
Think
to
about
actually
yeah.
truly identify that. But I thought that was a fascinating thing, and it was like it sort of pointed to where if I had been able to just lock myself in a lab and work on that for years, that's the direction I would have gone in. Anyway, let's move on.
Yeah,
That's
you got me thinking about wasps and bees and
yeah. So yeah, yeah.
prehistoric
So
times, and maybe you're right. Our immune system probably had lots of pressures to deal with this. There's something that you just said that is really, really cool. But
yeah, yeah. So I,
let's
I think yeah,
fast
because it all comes back to biology, etc.
forward.
Anyway, let's move on to this.
Pressure, right?
This is we've been building to this one because this is astonishing.
It
Well,
is.
it's not. Yeah, it's basically. You're probably going to do most of the talking on this one, but this is Philadelphia doctors treat infant with first custom CRISPR therapy that fixes genetic mutation. Now there's a lot of discussion within the pharma industry about personalized medicine in terms of oncology treatments, things like that. Where a lot of small molecules, oral therapies are just it's a treatment and everyone reacts differently, but that's the treatment. I know that in terms of Vertex and their cystic fibrosis treatments, they're particularly effective for certain mutations within within whatever causes cystic fibrosis. So that if you've got that particular mutation, you're more likely to benefit from treatment, etc. So it's like semi-personalized. This is truly personalized. This is so. Kyle Patrick Muldoon Jr. was born in August 2024, and they realised quite soon that KG, as his parents call him, was too tired to feed, his breathing was laboured, and he had sky-high levels of ammonia in his blood. Tested his DNA and found two typos that caused a rare metabolic disease called CPS1 deficiency, which affects fewer than 1 in a million newborns, and up to half die in early infancy. So the current treatment for it is a liver transplant, which could take months, and it's very expensive. A team of researchers at Children's Hospital of Philadelphia and University of Pennsylvania slowly started working on a cure. And so what they did, well, basically, the story of this is a 6-month sprint leading to a custom CRISPR gene editing drug to directly fix one of his two mutations, and basically which he's now been treated with. And the rising ammonia in the drug, I think if the ammonia levels rise above a certain level, even for like an hour, the IQ drops accordingly. It does highlight it in this article, and so watching that ammonia level is absolutely key. And so basically, they couldn't if this wasn't treated, having things like protein that spikes your ammonia, and so that like I said, there's a lot of effects on it. I'll pass it to you, and then we'll touch on more of this. This is unbelievably
John,
unbelievable.
it is, and just for time's sake, I'll keep my comments somewhat short. So you nailed it. This is personalized medicine on steroids. We probably didn't think this could happen like in our lifetime, right? That's how quickly this is advancing.
Yeah.
They started calling each patient like alpha, right? They had this particular disease. They removed the cells. They did what they called it like a boutique thing, but it was taking too long. It was too diverse. It had mutations. Well, they went through 7 iterations of this until they hit this particular patient. And it's so dense on how exactly they did this. But, CRISPR technologies, some grad student was in there and they had like, because of the timing of this disease, right? Meaning these children can die really young and they need to be treated quick. They thought that they had about 6 months to design this de novo CRISPR. John, that's
I know it's yeah.
Me, I'm laughing. I'm like, what? that could take a decade, right? So, but what they did, they used some liver cell lines. Again, this is where the wave of the future is. They used some AI. They called on multiple research institutions, right? So CHOP is the innovator of all this. CHOP is the innovator of CAR-Ts and let's call them cell-based immunotherapies. And Lou there is the guy who invents CRISPR. They're all coordinating together. They go out to the Berkeley. There's someone in Berkeley who's rapping on. Forgive me the names, but it's again that's the power of solving a problem is getting all these people with all this knowledge to collaborate together, and voila.
Yeah.
They ended up, making this Grandinetti is the grad student, and hats off to her. But then, without breaking down the entire mechanism, they've just reestablished this mutation, and now this child may be healthy, John. Right?
Yeah, sure, absolutely. Yeah. This is how incredible this is.
This is unbelievable. Yeah, go ahead.
Yeah, so this originated at CHOP, but I mean, UC Berkeley was involved. Is another, I think, Massachusetts General Hospital. I think that Benjamin Kleinsteiber gave the mechanism that had the successful editing.
Yeah, he was using the AI.
Right? And so it's just all these people coming together. I mean, like we sort of talked about the liver transplant being expensive. They haven't said about how expensive this is, but that doesn't really matter. This is a proof of concept. Added to which it could have cured a child and given them a healthy life. I mean, I think they say at the end of the article he will be coming out of hospital for the first time in his life at 9 months old, because it looks favourable going forward.
Yeah, I'm going to stop there. The mechanism behind it, just how to I can't tell if like it's a systemic thing that was changed, or is it just a given enzyme that might be in a given area, a given organ, right? That's still, but it's really I don't know. It's heartwarming, and just the rare disease space is so fascinating to both of us, John. That's the miracle of science, in my opinion. It's great that we can keep ourselves body by chemistry. I talk about that. I'm all in on trying to take things to make me live longer. But I feel like the treating of rare diseases and just the idea of like, how powerful is this? It's amazing. And I don't want to be a nanny naysayer or a downer a little bit, but I wonder whether is there side effects to this? It's still a little bit fascinating to me.
Well, yeah, I mean, this is so outside my realm. I don't know, but
I think it's outside of everybody's realm, to be honest.
Yeah.
I think some of what's happening, we truly don't understand. There's a gap in how and why it's all working, but maybe we'll move on. But I hope that made sense. Sure, you can draw it up on a chalkboard, right, on a dry erase board or on a computer and say, oh, this is how it's going to work. And then when it works, oftentimes it has nothing to do with what you drew up there, because the immune system and your body do a lot of wonky things. It's hard to determine if it's just this particular shot on goal matched this patient, but it's to be determined. If we're sitting here a few years from now and 8 out of 10 born with this can be treated with CRISPR, that's a huge success story. I mean, that is off the charts success there.
Yeah, yeah, absolutely. I think, yeah, like I say, I think it's the personalized piece. It's like this gives a direction. So, if you've got someone else with the same syndrome, is it the same treatment, or is it a tweak to what they've successfully done? So it's still like that personalized approach. What was interesting here is they did it as a 3-step treatment. So it's like one step to see, was it if a low dose to see what it was doing, then an intermediate, then a high, to sort of finally finish it off, because it was basically track and see how was the CRISPR functioning, was it affecting the symptoms such as high ammonia that I mean, they started off with still these other treatments to mop ammonia. They had the KJ caught a viral infection which can be life threatening for patients with this conditioning. A similar bout with the stomach bug caused his ammonia to rise when he was younger. This time he was fine. So it's like these signs that something was going right, and so it's the stepwise treatment and monitoring to make sure it works. I mean, it's just the whole process in terms of the speed they moved at, the thought process in terms of looking after him while he was going through this treatment, to sort of say this is successful now. Because then the next step, the last comment I think is, we've learned so much from KJ, and it would be a shame if we couldn't apply all the amazing experiences that we've had through this to help more kids, and that's definitely in the pipeline. We plan on helping more children. So it's really that KJ, for want of a better word, is a proof of concept. It's then how can we apply this wider to other people, and it is just absolutely incredible in terms of, compared to some of the things we talk about.
And John, it always takes thinking differently, doesn't it? Because they said,
sure, yeah,
the repeat dose strategy may defy the one and done goal of gene editing, right?
Yeah.
And then it's funny, I touched on it. This gave us the freedom. They say that we have no idea what the true safety profile might be. So that was
true. Yeah.
This kind of staged dosing thing allowed them to look at off target, and we've talked about what off
exactly. Yeah.
They still have some concerns, but it looks like it was a home run, and this individual may live a happy life. Hats off to KJ. So, John, let's switch gears and turn to our main portion. What do you think? Let's
go for it. Yes.
Yeah. So today we've got a very special guest. Jeff Goddard, our Senior VP of Corporate Development, is here. He's going to tell us all about the KCAS strategies for balancing growth with a good employee experience. And in general, just Jeff has been a key member, a key architect of our growth, specifically with mergers and acquisitions. So without further ado, Jeff, would you want to say hello again and kind of kick us off a little bit about your background and introduction and current role and so forth?
Sure. Happy to. Honored to be here. You guys have become celebrities amongst the bioanalytical world with the podcast. So having
a cult following, cult following out there.
It's an honor to be here. So super fortunate to have gotten on board with KCAS. It's been almost 4 years ago. After our investors bought into the company and wanted to work with the owners and the executive team and the management to figure out how we could become a bigger, more relevant player in the marketplace. And listening to you guys talk about all the amazing pace of healthcare change in general, and especially when it comes to different therapies and drug development, the modalities are becoming so complex and getting introduced at such a rapid pace. I think it's been interesting. I've been in the CRO industry for 25 plus years, and it's for a long time. I think you could be around you could be in a lab and kind of stay steady state for a while, but the last 15, 20 years, it's sort of become, you need to grow or you're going to die, I think is the short answer, because a lot of it is because the modalities and the need for advancements and new technologies and new people and new techniques is it's rapidly changing. And it's not just down on the front lines of medicine, but that goes all the way back to what we do, which is help in the R&D space before it actually gets out to the CVS and Walgreens shelves. And so management and the investors all put together a nice strategy to grow as an org. And part of that growth is, part of it's organic, or, building up our own internal capabilities, but some of that, to grow effectively and become more relevant for the larger, the mid-size and larger pharmaceutical companies that are doing now global clinical trials and such, we needed to expand. And one of those expansion options was to go and buy some labs that would fit well with us or expand our capabilities. And, one of my early remit was to go help lead that charge. Came out of this industry, spent some time in private equity backed BioA labs in the past. Had good fortunes to have some great experiences along the way, and locking arms with John and Amy and the team and you guys has been a heck of a ride. It's been a lot of fun, and hopefully the best is still yet to come. But so my role started helping on the front end with M&A, and so it was, I've been on the commercial side of things for quite a while. From a bag and a territory to BD rep to heading up global sales teams and things like that. So, and then ultimately was CEO at another lab at one point. So, my perspective on the M&A side was always bent towards operational, cultural fit and capabilities and things like that, versus, it sometimes a more traditional path to doing M&A work is a little bit more on the finance side. See a lot of people that grow up in the banking industry that can read a balance sheet a little bit more quickly than I can, but they tend to be in that space a lot. So I kind of came at it from a different perspective. Luckily, had a lot of help on the other side of it with our investors and finance teams and things like that to help. But yeah, we went. The quick rundown of what we did as an org was we came upon a wonderful opportunity in Flowmetric, which is one of only a couple labs in the world really that focus on flow cytometry. And I know you guys have done a lot of podcasts, and we know how quickly that space is growing in the cellular space, cellular immunology, and especially in the cell and gene therapy space, and just all sorts of applications. So Flowmetric was a lab that kind of got started from people that were at a large pharma company, kind of the flow core, and spun out and always had a vision to start a flow cytometry CRO. And we found them at the right time in the right place, and it was a great coming together. Really expanded our capabilities in that space, and it was in Philadelphia, which is, as we all know, it's sort of like, I don't know what they call it in the biospace map, but I think it's called Genetown, or no, that's Boston, but it's one of the it's a cell therapy mecca. Actually, you guys were just talking about the CHOP and the CHOP. I mean, that's ground zero for some of the latest and greatest technologies and innovations in the world. So we're fortunate to have a hub there.
All the rage is immune effector cell therapies.
There you go. Thank you.
So they're the mecca of immune effector cell therapies. So Jeff, thanks for that great background there. We're going to want to dig in a little bit more. I know you were on a little bit of a roll, but you started going right into business growth through strategic acquisitions and partnerships, right? And you mentioned Flowmetric, and you talked about, kind of like not many standalone BioA labs that did flow cytometry, but we were doing flow cytometry, weren't we? So maybe I guess my question becomes like, I often say to people, I don't know if it's a question, maybe it's more of a comment. I felt like we went out and bought the competition. So I was all in. I thought it was great. So maybe some experiences there.
I think my supplemental question to that is like, what's the process for thinking where do we go next, and then identifying targets? Is that working with the board, or how do we go about it?
Yeah, good. I mean, it's a fantastic question. So because it was a little bit before my time, but the plan came together of where are we now, where do we want to be, and sort of how do we get there. And where we were was a single site in Kansas, fantastic group group of scientists, fantastic reputation, not necessarily well known around the world as the huge BioA CRO. So one of the things was to expand our footprint strategically, and that meant potentially getting over into Europe and becoming a multi-site facility or multi-site laboratory, which fits generally speaking a little bit better with the ability to handle larger global clinical trials.
I've lost count of the number of conversations I was part of with my previous company. Like, you're not big enough because you're not international, and we need to have that global support before we'll consider you. So it's a really important thing to be looking at as part of growing an organization, definitely.
And Jeff, I want to add, despite having 39 years of 40 years of operations at the time, no one really still knew who we were. That's another.
Sure. Yeah.
Right. Not anybody's fault. It's just reality. It's just how it was. And so one area of strategic, focus was to expand and make sure we have an international footprint. And we did that we found a group called Active Biomarkers in Lyon, France, and they've become a wonderful, wonderful partner. Now our KCAS Bio Lyon facility. But you asked then, how do you then continue to determine what else we wanted to do? Another big strategic pillar that we were going after was to make sure that we were out ahead of technological development. And we do that at our Kansas facility fantastically, but the ability to sort of fast forward our flow capabilities and get out on, we did do flow in Kansas, no question about it. We probably did a fantastic job of it, but to grow organically, meaning, go hire people and train and become better and better and buy the latest and greatest equipment. That takes a fair amount of time, and you have to be out on the front lines, like building the reputation to get the opportunities. And very few companies want to be the first one to try something new with you. So the opportunity to effectively merge or buy a company that's already been doing this they have been in the marketplace for 10 plus years and had a wonderful reputation in the space and a very focused space of flow cytometry. So the ability to sort of fast forward our footprint and our reach and our recognition in that space was something that was, I don't know, arguably too hard to pass up. It was a great opportunity for us, and the cultures fit really well together. And we've invested a lot of money out there, and I think almost quadrupled the space. And it's a wonderful, wonderful growth story. Same with our European one. So if you think about international footprint, then adding technologies and capabilities to stay out on the leading edge is another one. And then, we did the same thing by investing in our own, in parallel. We've invested a lot of money and effort into growing our capabilities at home too, expanding the lab and moving into a brand new custom or purpose-built facility, as well as expanding our capabilities on both mass spec, LBA, molecular, the whole 9 yards. So,
and if I could step in, I think having that site in Europe makes a big difference. Again, you can be a lab with a really good reputation in the US, but it's interesting how little that translates over to Europe. We again, referring back to my previous career, that was exactly the same issue we had. We had a stellar reputation in the US, but we go to conferences in Europe and the UK, and people had no idea who we were, and it's like the Atlantic really sort of. It's sort of amazing how it sort of sucked up information between the two, So I was really glad to hear that we were getting a site in Europe and building from there, because knowing how critical that can be.
Yeah, maybe two things. For me, it's like the European piece. Before we had a European footprint, when people would ask us, did we have ability to process samples in Europe or overseas and things like that, I kind of just looked at them and said no, and hey, I'm going to assume we're not the right fit for you, right? I just knew it wasn't we were kind of centered to just U.S. trials, which for the growth we had or wanted was perfectly fine, given that that's where still good share of the dollars I think are spent in the U.S. But
Yeah.
and Jeff said it earlier, hey, if you're not growing, you're dying. That is kind of how the CRO space has moved. And then the other is I want to touch on the idea, because as somebody who was somewhat new to M&As and just in general, been at companies where it got bought or had things, but never really been part of the process until these acquisitions here. I used to think prior to it, hey, why don't we just elbow grease it? Why don't we just go out and do it ourselves organically? We don't need them, this and that. But man, when you think about, we had a concentrated level of flow cytometrists in Philadelphia that was no way we were going to grow that. It would have taken a decade to go out and get the talent we have there between, the Adams and the Christies and all those people we inherited, right, John? And even bringing on some of our own talent with Brian and so forth. But now we got 30 dedicated people doing flow cytometry over there, or 35 or something. It's amazing. And then what was Active Biomarkers? Their claim to fame was they had more biomarker platforms than any other CRO in continental Europe, right, Jeff?
Sure.
That was, when I got told that, I thought, oh, I can work with this, like it was me. But wow, that's a great thing. And we still capitalized on that. So I'm teeing it up for you. But there's another platform in there that you can mention. But I'll kick it back to you, Jeff. Appreciate some of that insight. But as I think a lot of scientists that are listening have the mindset I used to have of, well, heck, we're doing it here. We can just do it here. We can do it there. Jeff, you know how challenging that is to go out. You can do it. I'm going to be retired before we would have gotten to where we are with Philly and Leon.
You're absolutely right. I mean, so much of how you grow as a CRO is, and we have amazing capabilities. So it's always a balancing act of what can we do versus what should we do. And, we can do just about anything we want internally and grow and grow and grow. But it's always a balancing act, because it's, to take the profits we make and reinvest them in the company and grow that way. Sometimes it takes a while. And you get this, I don't know, sort of an inflection point where in order to grow to the next level, and there's these transition points. I've been in a lot of CROs, smaller time, you get into that, like, once you start approaching like 100 people, you can't remember everything on an Excel sheet and on your hands and toes that way anymore. And so you have to start to get systems in, and then you have to get bigger systems. And now we're multi-site. So now there's multiple currencies and multiple languages and all sorts of fun stuff like that.
Multiple cultures.
Different cultures. That's a huge part of the whole process and what it does in a wonderful way for us is, obviously we, not that we've been focused in the US forever, but we were physically there. So like the majority of our work came from the US. We did get samples from all over the world. No question about it. But, but getting different places and different labs in different areas has done wonders for us, just to help bridge a lot of cultures, because not everybody's traveled all over the world. And little known fact, I think we all know it, but there are a lot of pharmaceutical companies and a lot of wonderful biotechs that are located and headquartered elsewhere in the world. And they may have satellite research facilities in Boston or somewhere in the US, but the mothership, if you will, are over in Europe, and there's others around the world. And so it just gives us a little bit more opportunity to get to know our customers in a better way, and also help our internal folks with their professional and personal growth opportunities. It's just it's scary at first, because we're the human doesn't love change, and
scary. That's what it felt like at times.
Yeah, it does. It's really hard. It's like, and if you add on that, we're in this cluster of an industry which is super highly regulated, full of, it's change doesn't happen drastically fast in our world. So if you stack all those things, change in our space is hard. It's really, really hard. And I think that's where I was so impressed with our investors and everything when I was looking to come on board. They really put an emphasis on being people first, in our growth and doing it, what I use my air quotes, we're in podcast, but doing it the right way, which is really investing in the people. Obviously, we have amazing facilities, we have amazing equipment, but that's really just money. And the secret sauce that makes a CRO really special to work with and has the ability to delight customers and continue on the front lines is really the people. And their emphasis on that was refreshing, because sometimes you think that it's just all about the money, and it's really not. It's bring great people together and let them do great science. And when these pharma and biotech companies are giving us their babies, basically, I mean, their molecules to work with, it's a huge amount of trust and it's a huge amount of responsibility. And I think if I were on the other side, and you guys I think have been, you want to know that who you're trusting it with is going to do a great job, and they're going to be passionate about the work they do.
Man, Jeff, there's something. Sorry, I'll let you go in a second. Yeah. One thing we should probably be the most proud of as an organization is that I was part of the harmonization of both sites, and I don't think we had any loss. I think in general we've had all the people I started working with. The vast majority of them are still here, especially any that were in the bench or doing the work. It's something we should be real proud of. And I've been there a couple times over to Lyon. I've been to those sites, and it's because I've been on the other side of that, right? And some of my experience, where that's part of why I left. I never felt like I got embraced or a voice or whatever the site felt like, right? Those are the words. We don't have a voice here, things like that. We didn't experience much of that, Jeff, and that's something we should be pretty proud of.
I wanted to touch on that because that's why it's part of the topic for the discussion, and we've all been through mergers and acquisitions, and there's been some horror stories out in the industry. I think Pharmacy and Pfizer was noted as a particularly bad one, but that I think that to hear that the board are talking people first, that's what you want to hear, because, and so long as it isn't just paying lip service, because the harmonization between diverse businesses is absolutely key to the success of it. You can go out and buy businesses, but if you're not going to work with them to get to a common approach, then it's going to be a nightmare. So what was the approach for harmonization that basically made it successful, such that we didn't lose people? The impression I have, although I don't work particularly directly with Lyon, certainly not with the Philadelphia team, is it seemed we feel like a cohesive unit. I'm on one side, quite remote to those other teams, but we feel like a cohesive of company, and it doesn't feel like three different organizations.
Yeah, huge emphasis during the process, during the evaluation period, getting to know the people on the other side of the transaction, if you will, and then the real work begins when we actually come together, and that real work is the integration plans, and then executing it, and making sure that we have the systems, the processes, and everything to support us working as a, we call it like one KCAS. And
sure.
And so I've been a part of some that didn't have that exact approach. And it's hard too, because you've got different systems coming together. So sometimes you have to pick one and go with it. And, something that, I think one great example of, how important it was to us when we bought Flowmetric and Active Biomarkers, at some point we need to go through sort of the rebranding process, if you will, or the naming stuff. You can't just kind of keep Flowmetric a KCAS company for forever, and that's part of that transitionary period of bringing it all together. We spent a lot of time, and a decent amount of money figuring out what we were going to call ourselves, honestly, on the way forward. And I think we were particularly sensitive to, if we just folded into the purchaser, the KCAS at the time, we're very, very sensitive to how that might feel. on the other side. Well, here you go. Just stamp the name, call us what you want, and go. So, we purposely had people on the committee, if you will, on what we were gonna call ourselves, and we did a lot of market research. We had experts in this space helping us, because we wanted to make sure that whatever we came out the other side with the what's our integrated name and persona kind of going to be, is that we had full input from everybody, all sides, from the French team and from Philly, and so we honestly at the beginning we didn't think we'd be called KCAS anymore, because it probably would feel a little too over the top to force that name on everybody. And
it felt very Kansas City is what it felt.
Yeah, that too. I mean, and that was kind of a localized feeling too. I mean, those who have known us for a long time, right? The KC stands out. Now, luckily the Chiefs have built a nice worldwide reputation for us, so Kansas City is pretty well known everywhere. But
we settle on KCAS, right? KCAS, right?
KCAS, yeah.
Yeah. And what does that, magically, what does it do? It makes the Lyon and Philly site feel like, oh, we've got this new name. And it felt the Kansas City site like, oh, we get to keep our KCAS brand. Because people used to call us KCAS, right? Sure. So, I mean, that was kind of common. And to me, it sticks out because it's just kind of cool. But, right, that was the thought process there, right? It was a win-win all around. We as it turned out, we had a very positive reputation for those who knew us. Because of the involvement with everybody around the world on this project, we all came out of it with KCAS Bio, and all teams bought in, and it was the best solution for us on a go-forward basis. So it was that's just it's just one example, but we've got folks from each of those sites on our leadership team. You said it yourself. You've been over to Lyon. We move people around quite a bit and show up at conferences together. And we've got an integrated commercial team, and the scientific teams are all working together. One global quality system. I mean, it's all these things, kind of the IT infrastructure's there. So all these things are all ingredients so that when our clients go one site to the other, we're walking the talk, I guess, is what I'm trying to say. And we've also expanded that. And I know one of the topics, we've expanded that through a partnership down into the APAC region as well, to our Australian partner. And that was sort of a same sort of thing with a slightly lighter touch. We didn't go through a full acquisition, but we did a lot of work on partnering with the right group of people with the right facilities, so that we could continue to show as one cohesive unit. And I think that's been a wonderful success as well.
And I was just thinking about that before you touched on it. I mean, that where you're looking at setting up the same approach across sites has to build more teamwork between the sites, so it gives even more of that sense of being part of a unified company, because it emphasizes that you're in this together. And obviously Crux are part of it too, but to have Lyon and Philadelphia working directly with each other, it's got to be a positive thing.
And maybe I want to add a little bit here. The when we talk about people first here at KCAS Bio, that's why we were successful. It starts at the very top, even investors and executive team and yourself, Jeff, me, John, right? We all believe in the people. And so our harmonization between Philadelphia, what was then Flowmetric, and KCAS Bio, or KCAS at the time, that we learned a lot there. And we formed teams, and we had all sorts of real involvement. We took a real HR involvement, and we're going to talk to them on our next podcast about how they helped give us some ways to make sure we weren't, just collectively working together, both sides. And everything we learned from there when we went to Active Biomarker. And by the way, we moved all of our flow cytometry equipment over to Philly and all that. And so there was some great, it was really enjoyable for me personally. And then when we go to the Lyon site, we learn all the things we just learned. We were able to integrate really nicely. It was a lot fewer issues, or not that we had a lot. And then the other part is we actually built up their PK and ADA LBA work. So their biomarker knowledge is being influenced and transitioned over to us, and some of our biomarker knowledge back to them. But then our PK and ADA has really helped them. So that helped harmonize them. And you're going to talk about how we've done that because Watson limbs and stuff like that. But then when we add on Crux, it made it that much easier. That's what I'm getting to. So we were able to put equipment down there, right, Jeff? And that made it that, because we just knew we had a roadmap. That's the third time doing it, man. All of us got better, right? So that I know it was a little bit long there, but that was important to emphasize that it does start with, a lot of people talk about it, but we went out and did it, in my opinion.
Indeed. And it does get a little bit easier. And, and that honestly makes it easier on the other side too. If you're looking at, who do I want to partner with? And you're looking at, you probably have options as well. And so what makes KCAS a great partner? And, you get to talk to the people that came before that are still in Philly and in Lyon, and what's the process like? And, it is people first, and it's been a great approach. Now it's people first, but then the systems enable our great people to work together in a much more cohesive way. And that's investing in, like you said, Watson limbs. It's super important that
Meat Analytics, right, Jeff? Sorry, that Meat Analytics, what we call Holmes, really harmonized us as an organization, which is
absolutely
that we're only 90% done, right? We're still, harmonization is an evolution now.
Yeah, sure.
We're always still trying to harmonize. It's just,
continually.
We're like 95% of the way, but that next 5% is where, but we're growing together, right? It becomes even more of not a challenge. Just, I think it's exciting, but yes.
Yeah, it is. And it's a whole nother stage in our, long-term growth. But you mentioned it before about the HR and people getting in there. And I know you're going to have them on your next podcast, but that was another major part of our investors investing in us to grow what I again, my air quotes, grow the right way. Really put an emphasis on establishing a chief people officer and really formalizing and investing in the people aspect of it. And I think we've all worked at several places. I think we have the best group that I've ever been a part of, and they make the recruiting, the onboarding, the employee experience second to none. And it's been fun to see that evolve as well.
I think all the things that we disliked, we tried to avoid as a team, right, Jeff? All the things that rubbed us the wrong way, we talked about and ensured that we weren't going to let it repeat itself, because they're all the same. Even though, Jeff, whatever your experiences, whatever the downfall are, and John, whatever yours were, those things are universal.
Oh yeah,
There's some just lack of communication is what I'm going to say is what happened.
Yep.
Yeah.
Yep. I had some horror stories to relate, and yeah, we all do. Yeah.
Let's not open those wounds up though, because we're right on. I think we got a couple more agenda items. We're kind of, John, maybe you got something you wanted to add, but
I think this is good. I mean, I think you could roll the two in. I mean, we obviously we touched on infrastructure with Watson and Holmes, etc. I mean, it's that again. It's part of that.
Maybe on the IT side of things, Jeff. Can you talk a little bit about what Jason Kentner team's doing? Maybe John, we don't all see all that, Jeff. Maybe you see more of it, but we we get all sorts of exams, and you get put on a naughty list. I mean, we make a huge investment in IT, right? And that's only what I'm seeing. Maybe talk about that before we wrap up with our last item.
Well, and I think this would be the case whether we were still a single site or whether we were expanded. It just makes it more complicated when we have to deal with other rules and regs, GDPR and stuff in other countries. But nevertheless, the IT security is just paramount, and that's the best example of this. I mean, just take any services agreement we have with a big pharma company, and the section on data security and protection of data has gotten monumentally thicker in those documents over the last 10 years. And we all hear horror stories of, ransoms and takeovers and hacks and things like that. And so they're giving us the trust with the molecules, but what we end up producing is data, right? I mean, we bring a sample in from the backloading dock, and we send out a report at the end of the day, and or the end of the week or the month or however long those studies take, and that data is really what we produce, and to have it protected and secure, and the whole 9 yards around it is just it's table stakes, right? We can't be trusted with the samples if we can't protect the data. And I think
well, Jeff, I'll ask Maria. But I know it goes up through Maria. I think there's more I'm going to ask.
Yeah, but we've got a great group there too, and they do an awesome job.
Yeah, they do. But I think there's some. But anyhow, some more universe. Well, anyway. John, do you have anything before we move on to that last item?
Nothing. Let's just touch on the last thing. I mean, we talked about how we harmonize, and then we're always looking at expanding capabilities with new instrumentation, etc.
Exactly.
I mean, and what drives the decision as to who takes on a certain piece of equipment? Because I'll let you talk on this because it's all new to me. Dominic, if you want to drive it.
Yeah. Well, I think I'm going to. Jeff, so this system, right? It's the Nulisa, right? I believe we'll be able to say we can do PK on just about any compound, or at least biomarkers on any compound. But I personally, right, get a lot of instrument people coming at me, and we've been, I would say over the last 6 months to 9 months, Jeff, you and I have been interacting on a much higher level, right? So you're seeing all these things as well. And, you take a call, right? Because you want to learn, and maybe something's even creative, but a CRO has a real, I've gone through 3 or 4 instruments that have failed, been part of like GLP implementation of the Quinteric system. For those of you that didn't know that, KCAS Bio was the first to GLP validate that system. We helped them quite a bit back in the early days, although we no longer offer that platform. But I feel like we've been at the forefront of mass spec evolution, as well as things that happen in LBA, and all sorts of things that we do in terms of being able to look at an instrument, assess it, and even kind of kick the tires on if it's GLP validatable and so forth. So my question is, how did we get here with this Nulisa, and tell us a little bit about, because it's super exciting. Maybe walk us through the process of how we select an instrument and how we landed on this one.
Sure. So I love getting out. It starts with voice of the customer at the end of the day. Like and I think that's, one of my feet are in the, certainly been doing some of the M&A stuff, but another foot is on the commercial side, and helping build our strategic accounts and some of the big pharma groups, and helping our commercial team and our science teams keeping a finger on the pulse of what's the latest and greatest. And I think there's a distinctive difference between being on the bleeding edge of the latest and greatest, and being on the leading edge of it. And being on the leading edge of it is really where what's, again, one of our priorities as an org is to make sure that we're staying up on our abilities to handle all of the different modalities that are coming down the pipelines. And so it starts with customers telling us, hey, we're trying out these instruments. It starts with our own internal scientists going to conferences and keeping a pulse on what's the latest and greatest out there. It starts with you getting calls from new instrument vendors. And so we have an understanding of what all is out there. And then we leverage our great commercial relationships. Sometimes we even we can even pay for some research as needed, but we find out whether these things might just be kind of a one and done. It's nice to have, or are they needed to have? And is the general process, companies tend to bring some of these in-house, they play with them. And then ultimately, if the market's going to be there, they'll start to look for CRO partners that also bring these instruments on, so that they can start to outsource and support the clinical trials and some of their research efforts. So we kick the tires on both sides of that. How robust is the instruments, and we look to our internal teams to help evaluate. But super important is how much demand is going to be out there, and can we keep the machine busy? Can we keep the team busy? Because that's our lifeblood as a CRO, to kind of keep those things rolling. And every now and then you find an instrument that's really got sort of the best of all worlds. And we're really excited about the new LISA platform. It's both ultra sensitive and highly multiplexed, and the folks who have built it are industry veterans in this space, so they've put together quite an impressive instrument that just from over the last year of going to conferences and talking to our customers, we've got customers literally coming up and asking us if it's not every day that you get the "if you build it, we will come" approach, but it's a good opportunity for that. Do the same thing on the mass spec side. We've got one of the nicest, newest fleets, 7500 pluses, high-res instruments now. So we've done all that with a wonderful marriage of growing with our clients to make sure that we're out in front of not just what they need today, but also asking them, what are you going to need in 6 to 18 months so that we can look internally and invest in the appropriate places for instruments, people, space, et cetera, and make sure that we're gonna continue to stay super relevant to our client needs.
So for the new LISA, what's your principal application for that? I mean, what are clients asking for with it, and where's it? I know where's it being installed? We're
starting with it in Lyon because its primary purpose today is biomarker applications.
Okay.
It got started with the concept of doing it. They wanted to really tackle oncology biomarkers, but very quickly got pulled into the neurological space. As we all know, I mean, to find blood-based biomarkers for neurological diseases like Alzheimer's, Parkinson's, et cetera, is needles in haystacks, right? I mean, we're talking really low levels of p-tau and whatnot in the circulatory system, because not everybody wants to give their brain biopsies and CSF every day. So the Holy Grail is to find these markers that can really not only predict but help evaluate the diseases in the circulatory system. So anyway, that takes ultra sensitivity, and the Simoa instrument we still do have Quanterix. It's in the Lyon facility as well, but so primary purpose for new LISA is biomarkers, but there was a paper recently at WRIB, I think, that a pharma company has implemented it for PK as well. So we're really excited about the options for it, and it's got a development mode on it as well, so we can develop our de novo assays, and that's another important piece because we've got amazing developmental scientists that can develop novel methods. So it has singleplex, multiplex, and our ability to do our own development de novo assays on it as well. So it's really exciting.
Yeah, I want to brag on Lyon a little bit. They were doing LE spots and flow cytometry and all sorts of stuff. So taking on this platform, they got a lot of talent over there.
Yeah.
And this is a home run, Jeff, right? So I think I got introduced to it maybe 6-9 months ago, and I've been kind of hearing a little bit about it. It's kind of competing against Olink, right? It takes a lot of that sort of technology, but has like solved all the problems. It's quantitative. It can do up to like 250 biomarkers with a 25 microliter sample size. Now, you can't get quantitation on all of them. They're working on that, but they have certain panels, like up to, I think, like 35, you can get like quantitative, which is fabulous. Super sensitivity. It's like sub-picomolar, femtomolar detection levels. But the other good, you mentioned the talent there. I was pleasantly surprised to learn about that. They have a very reputable individual leading their business development team, right? And as well as I was happy to hear a couple other people have already GLP validated it, because I don't want to be the first anymore. So I was pretty happy to hear that. So this thing, that's why I think it's a home run. And we're going to launch it in Lyon, as you mentioned, Jeff, and then we'll see the sky's the limit from there. But I mean, of course, we're going to want one over here in the U.S. I mean, that's just a given. If it performs as advertised, which it appears it does, they have all the people from Illumina's and places like that. They got all the right people that kind of took on kind of filled a void. This is pretty exciting stuff. It's very exciting.
Indeed. And, it's, for us, the contrast us because physically we can keep a cluster of a variety of instruments in one location. The typical biomarker can be, it's plasma serum based typically, so you can freeze and ship and still have a super robust assay. Flip the switch over to our flow cytometry, where we took spectral flow. We did that whole business case in a very similar way, voice of the customer driven, but in flow you've got to be regionalized so that you can get fresh samples within hours or at least within days to a local facility to do the work. And so when it came to flow, it was absolutely critical for us to, as we expanded, to put harmonized instruments in all three sites, including the U.S. plus Europe plus our Australian partner, and that's because you have to have relatively localized ability to handle those fresh samples. So different instruments, different needs, similar approach in letting our customers help drive the needs. So sure. we should hopefully we can ship samples to Lyon very, very easily and do a ton of biomarker work for customers from anywhere in the world. And
they have the Ella system
and that's the flow cytometry samples to any of our locations.
Sorry, Jeff. So we routinely do that, right? So they have an Ella. You mentioned the Quanterix. We don't have those here.
No, Ella, Quanterix, LumiPulse, which is another incredibly novel biomarker platform, and the Simoa, Luminex, MSD. They've got the whole 9 yards. I know we definitely have MSD and Luminex in the Kansas site, so there's a lot of harmonization there. They do the bulk of the PK work, but when it comes to a wide variety of biomarker platforms for just about any application, it's hard to find a site better prepared better capable than the Lyon site.
I believe in continental Europe they have more biomarker platforms than any other CRO.
Yep, that's what.
I wonder if we could figure that. I'm sure I would bet on it, but something to research for you, Jeff. No, just kidding. We're winding down here. Pleasure having you, John. Any last questions?
No, it was a real pleasure to have you join us, Jeff. We've alluded to it, but we have a couple of guests next time. We're touching more on this from a couple of different perspectives, and we'll leave that for now, and we'll introduce them properly come the next podcast. But this is a key part of as to what makes KCAS who they are, in terms of how we're growing and keeping it, making sure it's successful for the people as well as the business. Shall we move on?
Yeah, so we're gonna put you in the hot seat, Jeff.
Hey.
We're gonna go with what's your favorite podcast episode? Although I think I could guess it. You got one for us?
What comes to mind, one of my most entertaining ones to listen to you guys, it was probably the webinar that you did with LBA versus LCMS for ADCs and stuff like that. That's one of my most. I could watch that on repeat a while, but that's not a podcast, I don't think.
No.
Probably the best podcast. Something I loved was the EBF recap, because I think that was it was after the first time you'd been to that conference, right, Dom? And listening to your just like initial impressions of that wonderful conference was fun to listen to, because I felt the same way the first time I went quite a long time ago. But I know it's easy to see why John is so passionate about it, because it's just a phenomenal group.
Yeah, he's like a rock star over there.
He is.
I was going to guess the GLP-1 episode.
Oh, yeah.
Wasn't sure if you had heard those or not, but I thought that might be near and dear. But we might have you on the next time we do one of those. We kind of touched on that offline a little bit. But you maybe John, you want the next question? You want me to just?
Sure, favorite dish to make. Let's go through the list as usual.
Ooh, I love to cook. I don't do it as often as I'd like, but I guess the latest thing I've been having fun with is just full on surf and turf, like a nice steak and some form of seafood. I've done lobsters, scallops, shrimp, and either do a fillet or a nice ribeye. But what I've done lately is my brother-in-law got me a sous vide, the French cooking water bath, a couple years ago for Christmas, and I've started using that to cook the steaks. And oh my goodness, it's amazing, absolutely amazing.
So, John, I've talked about my sous vide. It's precision cooking. I love the sous vide. Have you evolved into getting all the fancy coolers and tools and housing units? Have you gotten to that point yet?
No, I've got a big plastic bucket or the tub and the basic rack and just the regular thing. But definitely.
But you got a sealer, right? And all sorts of.
Yeah, I do got the vacuum sealer and the.
Yeah. So do you sear them when you're done, and then we can go on to the. So you're right. A sous vide steak is the best. And by the way, if you sous vide a steak, you can take them to a tailgate and make steak.
Oh,
because all you got to do is sear them a little bit, right? And
yeah. Oh, that's brilliant.
Yeah.
I got given one for Christmas, but I haven't broken it out yet. I'm still on the traditional cooking methods as much.
Yeah, John, it's funny. I had mine for like a year.
Yeah.
My wife was just getting on me. She's like, I bought you this thing because I thought you'd like it, and now I love it. So, but it was as soon as you get into it, and it's not that it's intimidating. The first time you do it, it feels like a lot of work because at least I thought it was because I'm managing all these things and like you need an app and stuff. But anyhow, let's move on to our before we start talking about our weekend plans because it is a big weekend coming up. Actually, Jeff, what's your favorite beverage? Alcoholic beverage.
Oh, alcoholic beverage. Yeah. Same either way.
Don't tell me you like Kool-Aid, Jeff. We know you like the Kool-Aid.
Yeah, I guess my latest kick is in the bourbon space. Definitely got. I love wine, but yeah, probably bourbon is my go-to these days.
You're liking? Is there a bourbon?
I'm not super picky. I definitely appreciate the nicer ones, but like in my collection, I've got Woodford Reserve, I've got Basil Hayden, and I've got Angel's Envy. Are kind of that's when I go through Costco and drop.
I thought the best on that list. But it's interesting you say that. I actually a friend of mine started up a monthly bourbon whiskey tasting.
Ooh.
And we did month 2, but you got to come with notes. And like I had some notes, but these guys were taking it super serious. So I was like, man, I better get ready next time. And there's like
you learn a lot.
Yeah, it was fun. But John, any comments there on?
I'm still on the single malt side of things, so I'm slowly working my way through distilleries I've never come across before. In actual fact, I found out just last week I was born in the south of Scotland. The first town I lived in was Annan, which is west of Dumfries. Dumfries being like the main city in Dumfries and Galloway. And it turns out Annan has a lowland whiskey distillery. It had one years ago. It shut down and it's just been revived. And so my younger sister lives across the border from Dumfries and just by Carlisle, so right on the Scottish-English border. And they've done a bunch of distilleries, and I was like, have you done Annandale yet? And they haven't. So I'm like, I think that's one I need to research and try and figure out if I can add that to the list. But yeah,
you've been to Scotland down. Have you done anything like that? Or right, you've done like a trip.
I've never done any distilleries actually. I just drink the whiskey. The closest there's actually a pub in Glasgow. I think it's called the Pot Still, that specialises in whiskies. So you go in there and you're really strongly discouraged from drinking beer because they got so many whiskies there. So, but I've never, I had a distillery right on the edge of the village I grew up in, but I've never actually been to any distilleries. It's a gap because I know I've mentioned this, but one of my friends from school is now a guide at Glengoyne.
You did mention that.
Yeah, and then actually one of my other friends from school used to work for Auchentoshan and other places. So I have my links to the whiskey trade. I just haven't taken advantage of it yet.
It's funny. That's on my bucket list to do it in Scotland, but there's one here in the U.S. and that's on the bucket list as well. The Kentucky one is quite quite famous as well. Jeff, you ever do any of those yet, or you've been to the?
I've I've been to the Guinness factory, but that's not obviously not a distillery. But we went to the United Distillers and Vintners, which is the big. They own a lot of different brands, but when I did a economic development trip over to Scotland, Ireland many years ago, and that introduced me to the single malts and the whiskey in Scotland. So that was my first. I'll say that was my first brown liquor experience, and I'm still a big fan of the single malts. I like the Islay ones that are nice and mossy.
Yeah, I'm only just touching my feet on those. So I found when we were in Edinburgh last time, we stumbled across Bunnahabhain, which is only lightly peaty. It's their 12-year, and it's like that's a lovely whiskey to drink. So I haven't really got into the full-on peat monsters yet, but that'll happen, you know.
I don't love the full peat throttle unless there's some heat. I actually like a lot of heat with it.
There you go.
So.
Heat, but
one more question. Yeah, what are your weekend plans? Since it's a big
Memorial Day, right? Big Memorial Day.
Well, there might be some bourbon involved, I'm sure, at some point. Actually, this is the Memorial Day weekend, right? So big things here. I live in Indianapolis, so it's race weekend. That's kind of a big deal. I don't think I'll be going to the race, but certainly we'll be watching and celebrating. But
right? Does it
so
at that time?
Yeah.
So I mean, how involved does Indianapolis as a city get going on this? Because I'll relate. When I lived in Cardiff years ago, whenever there was a rugby international, I don't know how big the ground was that held, the fans to actually see the rugby, but the whole city would go out and get drunk together. It was just the most unbelievably brilliant experience. I've never seen a live rugby international, but I've celebrated a number of them in Cardiff and had an absolute blast doing it.
Well, the Indy 500 is somewhat unique in that the cars are here for multiple weeks, and so they've been practicing and tuning everything up on the track. So it's an event you can go down to the track just about any day of the week for the whole month of May and watch them warm up the cars, do their test laps, walk through the pits, and see really interactive experience. People come from all over the world to be a part of that.
Yeah, I've seen testing there once, but years ago, but it was just a one-off.
It's a lot of fun. It's really a neat experience, and the city itself, this whole region, kind of I won't say shuts down because it really builds up, but there's checkered flags everywhere, and it's definitely a big deal. I'm not originally from here, so it's not like part of my heritage growing up, but it's easy to jump on board. But also that with a whole lot of college softball, which is a whole other end of the spectrum. But I think you guys know my oldest daughter played in college, and my youngest one is in high school and is really good and fun to watch. And so having three girls myself, and I'm deep into not only the ballet world with my middle daughter, but the softball world with my
you're doing excel. You say it so casually. She played for Duke, right? And did she play in the College World Series as well, or no? Did she?
She did. Yeah, she got to play in the College World Series last year. It was only a year ago. I was out in Oklahoma City about this time. So
unfortunately, I'm rubbing right up against my time limit here. John, maybe quickly tell us a little bit about your weekend.
I don't have much planned at all. It's going to be I'm taking the day off tomorrow to clean out of some storage stuff, so helping my wife with things. But it's just going to be a nice, relaxed weekend. I think weather isn't going to be great here. We're looking at next weekend for the decent weather.
You're advanced in age, but taking a day off to clean stuff up is something you look forward to, actually, right? You're excited for it, right? So that's kind of cool.
We're slowly rationalizing things. We've got a bunch of stuff in storage all over the place, and we're trying to reduce that because, when parents passed away, a lot of stuff came over from the UK that we couldn't fit in the house, and now we're trying to figure out a better way of accommodating things. So this is actually a big deal for us, you know.
Yeah,
And plus we'll like use it as an excuse to go for lunch and have a nice drink and everything. No, it's not going to be just. It's not
there's no time. Yeah, I got you. There's no, like, if it doesn't get it fully done or you get to X and Y, you'll be okay.
Exactly.
I've got I'm actually doing some dinner plans with some colleagues from work here locally. We're gonna go out for some Mexican food tomorrow night, and then Saturday night, my neighbor's having a little barbecue. We're going to join them. And then my son's home, so we'll spend some time with him and maybe do some landscaping. But it's a casual one. No, none of us are doing anything fun like going to a lake or doing any of that, which is usually what we hear about this time of year. But anyway, Jeff, thanks for joining us. We'll see you in the funny pages. John, you want to wrap this up?
Absolutely. Thanks very much, Jeff. It has been a pleasure. And obviously, we'll look and see if we can have you back again in the future.
Been an honor, guys. Appreciate it.